Going to be a answer key video for our first exam, and we've already experienced a little of that with our couple of quizzes we've had. So this will be an answer key, and I'm going to talk through the exam. And I don't write very many comments on student exams because I think if I have some coaching or help on answers and what are the good answers and what are not so good answers, everybody should have access to that. And so let me start out by just mentioning how I grade, because this is the first example of a big assignment where you can see how I grade. And what I always do when I grade is, after I grade the scores on this test, I make an anonymous distribution of scores and provide that as a handout, so you can see how you did and how everybody else in class did. You don't know who anybody is, so it's completely anonymous, and each student is represented just by a little asterisk. So let's say that you had an 81, and that was your score, and that represents you. Maybe two people had an 81 on the exam. And then next to it, I give a letter grade, and I don't use a 90, 80, 70, 60 cutoff, and I don't ever curve scores or anything like that. I just look at these scores. I've taught this course many, many times, and I say that based on what I just graded overall, I think students that got an 81 should get a B. And it turns out my grades are similar to the 90, 80, 70, 60 cutoff, but my exams are hard. We've covered a lot of material. I think the answers that we'll go through are straightforward questions, but it's just a lot of information. So I can't really expect a student to get them all right, or even get them all in the 90s, right? We have a lot of 80s and 80s on down like that. And so you'll see, I'll say somebody that's got an 89, well, in some cases I'll say that's great, and I think they should get an A or maybe an A minus, like that, and I based it on that exam. And so for each exam, I have a new distribution, grade distribution. You can see that. You can see, you got a B. Why did you get a B? Well, because some students got a little bit higher score, maybe got a B plus, and then there's a student up there that got an A minus. So the grades are always ranked like that, and we can discuss your grade. The next thing I want to mention is students don't do as well as they want on the first exam. How can you do better? And I've said this many times, is happening in class. There are very few points on our exam that are straight out of the book, and we never talked about it at all in class. And I'll talk about those. We're reading the book because that's how challenging biology is. But I want you to remember, really learn this stuff and remember it, some of it, lots of it, for the rest of your life. That where triploblast is something that you should remember as well as you remember that we have arms and legs, in my opinion. So I'm trying to teach the course that way, so you have access to this in a way that you will always remember, that water is H2O. Those are really valuable pieces of knowledge, and this course is just chock full of it, because although it's an advanced course, it's an introduction to this huge area of biology. So let's talk about your grade and where you're at, and if you want to do better, big focus is on that. And I'll talk about that in class as well, because if something gets brought up in class, then it's fair game for the exam. And so you want to take notes, you want to organize your notes into a study guide, because that's the most important stuff for you to learn about. And then you want to dive into the textbook to help it, use it to help you understand what we talked about in class, then get more stuff out of the book. And I would love to talk about more with you individually, or in class. Ask a question in class about that. And so I'll try to go through this efficiently. What is the term for a genetic factor that cannot be rescued by the father? So that's the official term that I prescribed for this. That's the official definition for this term, maternal factor. So maternal factors are not simply early embryonic factors. They're factors that mom made and put into the egg. Every animal on our planet has a big egg except mammals. Every animal on our planet has mom making that big egg in her body before fertilization. The egg matures, and it's full of nutrients and building blocks and also maternal factors. People don't think that mammals have any maternal factors. And so there's a description, there are early embryonic factors, that's a description. The actual definition is a really good one because this is actually a genetic term. And it's a term that in this embryo, if there's a defect in the embryo, it might be caused exclusively by maternal factors that were required from mom that couldn't be made in dad. Okay? So these are all examples of this TGF beta family. And where did that come from? It came from that handout. We learned that VEG1 and XNR are TGF beta family members. And then we didn't really learn about BMP4 yet, but this is also a member of that family. And there was a table in your book that we pointed out that for our exam, we were just focusing on 3 families of signaling factors, and signaling factors are very important, so there better be lots of questions on here about signaling factors. And the signaling factor families that we've learned about so far is TGF beta, just a little bit, and then sonic hedgehog, also just a little bit, and Wnt, a little bit more. Okay, so that's where that came from. And what are signaling factors called if they only diffuse short distances? This is what we talked about in class when we introduced when we introduced signaling factors and gave an overview in the context of the Wnt assignment. So I think it's probably mentioned in both the Wnt video and also in our class. I'm not sure if the book uses this term. And so there's plenty of that, plenty of stuff where we only talked about it in class, and it's not in the book, not because it's not important for the field, but for one reason or another, it just doesn't pop up in the book, or it might be in another, section of the book that we're not, we haven't covered yet. What region of the mesoderm gets hollowed out? And that's the lateral plate mesoderm. So that we talked about many times. There's the notochord, there's the pairs of somites, bilateral somites, and then there's that lateral plate mesoderm. It gets hollowed out to form both the somatopleura and the splanchnopleural lining of our coelomic cavity and coelomic cavity in all vertebrates. How are members of a gene family created? And that's this type of mutation that's called a gene duplication. And I gave answers. A lot of times I'll put down okay. If I write okay in your exam, it means like that wasn't what I was looking for, but it's an acceptable answer. So because at least one student said, how are members of the gene family created? And they might say they were made by forming paralogs. I don't remember if that specifically, but I know there were a couple answers that weren't exactly gene duplication that got credit. RNA interference can block gene expression in 2 different ways. Not very many people got this right, and I thought it was, so it's interesting for me to see, and it's helpful for me to see, which things students seem to understand and which they don't seem to understand. A lot of times it might just be the way that I organize the way I've described it, but because the answer to this is that either the RNA interference works by blocking translation or by causing RNA degradation. The mRNA gets destroyed. And so that's that one. And reasons on here is because we talked about RNA interference some, and although it's a detail, the Nobel Prize was given this year, so we spent 10 minutes in class talking about the distinction between Nobel Prize given in 2006 and 2024. And again, I don't care, that you know anything about the years or the people involved, but there are actually 2 different mechanisms for RNA interference to occur, and those are the 2. What happens to a cell when caspase is activated? And the answer is programmed cell death, or you could say apoptosis. So I'm very open, to, well, you didn't have to exactly say programmed cell death. You could say apoptosis or death. That would be great. What part of a gene is transcribed by RNA polymerase? This was the transcription unit. A few students put down coding region. Coding region is wrong. Why is coding region wrong? Well, the only genes that have a coding region are coding genes. And we emphasized a few times in class there are 2 types of genes. One type is coding gene, and the other is non-coding genes. So that's the first reason that coding region is not good. The second reason is that coding region is just a part of the transcription unit. So there's the transcription unit, and the coding region is a subset of the transcription unit. It seems like a good answer, but part of a gene is transcribed by the RNA, but that's kind of an intro-bio answer, because in intro-bio, the big emphasis is on, oh, genes code for proteins. Well, many genes do not code for proteins. Like the genes that code for microRNAs and small interfering RNAs do not code for proteins. They do not have coding regions. So this is from the early quizzes in our course. And so Darwin didn't have phylogenetic evidence, and instead he used this morphological evidence. So what you can see, some students wrote down morphological, and that's a fine answer. The term that we used is phonetic. And it's great. There's no criticism of Darwin. It's just that it is true that he didn't have phylogenetic evidence. Phylogenetic evidence is considered modern DNA-based evidence, where you can say, look at how closely similar these DNA sequences are. Let's build evolutionary trees based on DNA sequence analysis. What does the notochord induce? And the answer is the neural tube, or the central nervous system. And that's that. It was a general term for mutation. These are called homeotic mutations or homeotic transformation. The example we've had so far are the Hox genes. The Hox genes are genes that are homeotic genes. When mutated, they cause a homeotic transformation along the anterior-posterior axis of any animal that has an anterior-posterior axis. What morphogen patterns the identity of each of our fingers? So this just came up briefly in class, but it was also described in our textbook in the very first chapter. So it's Sonic Hedgehog, and the emphasis in our textbook so far has been on the role in the development of the vertebrate face. But in class we said that this Sonic Hedgehog is a morphogen, and that's a signaling factor that causes different responses in target cells based on the signaling factor's concentration. At implantation, human embryos are composed of two types of cells, the trophoblast or trovectoderm and the inner cell mass. And that came up because we said, let's not worry about all the details the book puts in about mammalian embryogenesis and the development of extra embryonic tissues, except let's really appreciate that at the time of implantation into the uterine wall, the early human embryos and mammalian embryos in general have the embryonic stem cells are starting to become limited in their capacity, and so they're no longer considered embryonic stem cells. What cells migrate from the neural folds to form our face? Those are the very famous neural crest cells. And we talked about those a few times, and we'll talk about them more. What is this called? It's called an atavism, an evolutionary throwback, and where it's a mutation or maybe just an environmental disruption that causes an altered body that has a phenotype with similarity to the ancestral form of the organism. The receptor for Wnt is frizzled. Then I'm just jumping ahead to these because these are also some short answer questions. And so we have this drawing, and it's going to ask about these stages, and then it's going to ask about what the embryo is called during those stages. And so the stages are going to be fertilization. The second stage we talked a lot about, and students just, I think, kind of forgot that that's the stage called mitotic cleavage or cleavage. Several students got that right. Some students wrote blastulation because the next one is gastrulation, and after that it's neurulation. So those are the stage or the process that's happening. And then what this is called is the first one that's called a blastula, hollow ball of cells. The second one is called a gastrula, when morphogenesis is beginning and cells are starting to move into their proper position. And then the third one is called neurula. Great. Okay. And then there's this one, and this is based on that handout. And one of the things I should have mentioned at the start is, and I'm sure I'll mention this in class too when I hand back the exam, is on every exam, students will have studied an individual student will have studied a particular set of information, and it's not represented the way they thought it'd be on the exam. And one of those things is like this previous thing, where it's just drawing. And I could ask this by showing you image and asking you to fill in the blanks, or I could say just to explain what's seen under a microscope using those terms and what's happening. Either one of those is great. And in this exam, I just didn't have any long essay questions, but, and here's a key idea, the things I didn't ask on this exam in detail, I can ask on the final, because the final covers everything. And so if there are questions that you really wanted to answer on this or the second exam, just send me an email and say, I wish that would have been on the exam, because I'll put it on the final, and because the final covers everything. Great. Okay, so this one is really about the handout, and I was drawing this to help explain what this means. I didn't show all of them, but the idea is that you would understand based on this example that I did show in class what this would look like. And so disheveled is a maternal factor that starts out down here, and then after fertilization in the blastula, some of it is moved up here. Now you could show it down here still because it's still down here, but this is the stuff that's going to be the relevant disheveled. It's in a new spot, and it's left the inhibitor behind. And it's important to appreciate that we're showing the protein. So the protein's already there. We don't need to have it made by the embryo. Protein's already there. It's a maternal factor. Then beta-catenin is everywhere, and so I drew this out in class. It's just everywhere, and it's a maternal factor that's loaded into the egg everywhere. But then, at the time of the blastula, it's going to get degraded everywhere except here. And so when occurs, this beta-catenin can do its job, and beta-catenin's job is to overcome Siamoas repression by TCF, and then Siamoas protein will bind to glucosid, and glucosid will be expressed here in the embryo, and then it will bind to and activate genes like noggin. So Siamoas, glucosid, and noggin are not maternal factors. They need to get made in the embryo. These, if dad provides good alleles for these, even if mom's mutated, the embryo could be normal. And so then, let's see, did I miss one of these? So here's glucosid, and I just mentioned this. Glucosid is not present in the egg. It's not a maternal factor. It is present in the blastula, and it only gets activated here. Of course, the gene is everywhere. I'm not asking you to draw the DNA, the gene. We're asking you to draw the protein, and the protein is only here because that's the only place where beta-catenin can overcome repression by TCF and get the gene, glucosid gene, activated and transcribed into RNA, and then the mRNA translated into protein. I haven't used it in class, but in contrast to maternal factors, so ones that need the mom's genome to get the job done because they're done before fertilization, glucosid is referred to, and so this is a new term, zygotic factors. So that's factors that get expressed in the embryo. And those zygotic factors are expressed both on mom and dad's genes. So even if a mom is mutated, maybe dad can rescue or play this role for glucosid. Then VEG-T. So your handout says VEG-T is preloaded as a mRNA. So there's no VEG-T protein in the unfertilized egg. Then it gets made. It's a transcription factor, so it doesn't leave the cell. Lots of students thought it would move up here. It doesn't. It's going to activate genes that do move up here. So some students drew some arrows like that, keeping VEG-T protein here, but having downstream target genes expressed that are signaling factors that will leave the cell, like TGF-beta family is like members like X and R, nodal related genes. And the X is for Xenopus, the frog. Then brachyury is also a zygotic factor. It's not expressed as a maternal factor, so none here. And then it's expressed in the whole belt. It's a mesoderm specifier, and that's a big goal of the handout, is to activate mesoderm and mesoderm expression. So on to these essay questions that take a little longer time to describe, but I tried to make them direct and try to help guide you toward what I would look for in an answer. So instead of just saying, briefly explain the importance of induced pluripotent stem cells, I tried to guide you to explain some of the relevance of the these terms in that context. And so the first one, genomic equivalence. So why is this relevant? And the answer is that genomic equivalence means that every cell has all the genes. And that's relevant because it might be able to become an IPC. So why is this relevant? Well, it's because cells have all the genes, and that's necessary if you're going to turn a cell into an IPC. Then these all are built on each other like that, and some students appreciated that and turned this into a little essay. So they have all the genes, but in terminally differentiated cells like muscle, brain, skin, the terminally differentiated cells have stopped dividing and have methylated many of the genes they don't need. So yes, they have all the genes, but to make an IPC, you're going to have to remove this DNA methylation and make this cell so it's no longer terminally differentiated. Then this next one, I was hoping you'd appreciate that. The factors that are the stem cell identity factors, also called the Yamanek factors, they're all transcription factors. So to change any cell into an induced pluripotent cell, we're going to need to change gene expression. Yes, the cell has all the genes, but it's going to be some transcription factors that play the central role in changing the cell. And then finally, one of the features that's going to have to change is terminally differentiated cells have stopped dividing. We need to get the cell cycle going again. Great. And these are open-ended questions, and I have to be sensitive to the variety of answers I get and whether they're doing a good job. So you'll see, students had very different answers, and they both might have gotten full points because they kind of emphasized a different feature of genomic equivalence, for example, that is significant in the context of the induced pluripotent stem cells. Describe the terms fate, specification, determination, and explain the terms. So here's something that nobody did, which is true and was explained in class and in your textbook, and that is these are terms from experimental embryology. They're all experimental terms. So fate is when you tag a cell and observe a normal embryo, what the cell becomes is its fate. Now, students got credit because, they did a good job describing fate using other words, but that's actually the correct answer. And the same is true here. Specification is when an embryo cell has developed to the point where if you remove it to a neutral environment, it will reach its normal fate. Before that, it doesn't reach its normal fate. It's unspecified. Determination is if you move the cell to any other location in the embryo, it will still reach its normal fate. It is no longer sensitive to external signaling factors and the different microenvironments of an embryo. So that's why those all came up on that one figure, and it was in chapter 1. And it would be interesting for me to go back and point out all the points on this exam that were rooted in that chapter 1. It's just a big chapter that we built on. And some students describe specification based on these experimental procedures I don't know that anybody did describe determination very well. And so students lost a lot of points on this, um, even though I was giving credit um, for answers that were imperfect, that were, explaining things properly or appropriately, if not properly. And, I just have to do my best job. And what I do with each of these questions, I haven't mentioned this before in class, is I grade a question, I grade these one at a time. So they're anonymous, and I look at this question and I grade everyone's question number 2. I'm making comparisons between them and thinking about, like, I guess I didn't explain this clearly enough, that these are just all experimental terms in class. And so I have to be sensitive to that, right? Because even though I think I explained it, I guess I, didn't put enough emphasis on it for students to really appreciate that that's what's going on. They're all in the same figure, and they're all terms related to the fate of a cell. And then the next one is draw a simple picture of a somite and the sources of the 3 morphogens that depend on the somite in a dermatom. Label your drawing with the names of the 3 sources and the morphogen each secretes. So let's just go over here and, do I not have this set up? Oh, I don't have this set up in a way to flip over to my drill drawing board, so I'll put it right here. So there's the neural tube, there's the notochord, here's the somite, and the sources are the top of the neural tube, the roof plate, the bottom of the neural tube, and the notochord, and the lateral plate mesoderm. So 3 sources to label, that's a somite. And then made in each of these 3 sources are some wince, and some sonic hedgehog, and some TGF beta. So back to that, early question on the exam about what TGF beta is. We learned about these 3 different famous morphogenic signaling factors, or signaling factors that can be morphogens, and there they are. And so this was a figure from chapter 5 that we picked up. And so, when I say, look, let's learn these figures, then they're important. We got to really understand what they're out there. And I explained why they're out there is because this just provides a really satisfying way to see how each of the positions in the somite has a different physical address, where these guys are going to see a lot more sonic hedgehog than these guys. And that's going to affect their fate. So these cells are getting specified by the signaling factors. And so that's that one. Then scientists use genetic screens to discover genes involved in programmed cell death, but it's got the following terms. And so combination test cross, I think people got that right, which is, well, to do the screen, we need to do some breeding to determine if our mutant strains are in 1 gene, so different alleles of the same gene, or 2 genes. And if they're 1 gene, they will fail to complement. And if there are 2 genes, they will complement or rescue the mutant embryos. Invariant cell lineage is a feature of C. elegans. It's an important idea that it's not found everywhere, and it's that each embryo develops the same pattern of cells. Caspase is a protein that causes apoptosis, so they found the gene for caspases, right? So it's a genetic screen. What did they find? They found the gene for caspase, and caspase protein causes apoptosis. So when they found a null allele of caspase, there was no programmed cell death. Second site suppressor, everybody got credit for that. That was a question that I forgot that we didn't get a chance to cover. It's a genetic term that we just didn't go into depth a little bit more and get that covered. And I thought we had, but I realized, as I'm grading, I'm like, yeah, no, we didn't get for that. And then a null allele is an allele that has 0 activity. Completely destroyed. Okay, on to these, and then we'll be all set. So, humanized mice, and we talked about those quite a bit, and talked about some different types of transgenic mice that we could make. The first one is a model organism, and most students just said mice are model organisms, and that's true. But what is a model organism? And in our course, the answer is they have orthologs. That's why we can talk about a model organism rather than just mice. We're studying mice. Well, we're studying mice because they are related to people, related by common ancestry. The way they're related that's significant for experiments is orthologs. So in this context, there are some, for this humanized mice, we use the example of the liver. And the liver has all the orthologous genes in mice and humans, but there's slight differences. And so a drug might be metabolized slightly differently. So we're going to replace this mouse liver enzyme ortholog with the human to make it perform more like that. So identical twins. And and there's several things to say about that, but the most important one I thought is that scientists found that this natural splitting of a human embryo into, or a mammalian embryo into two, that could be done in the opposite way, where we take two embryos and we put them together and we get one chimeric embryo. Ortholog, well, we had that, right? Then you should just say the definition, because we emphasize that definition on a quiz, and we want you to be able to say the definition. And the definition is two genes in different organisms that share a common ancestor. So you would say two genes in different organisms, say they have a common ancestor, and for the liver genes, there are orthologs, the mouse gene, the human gene. Then transcription unit. When we want to replace the gene, what we would replace is the business part of the gene, the transcription unit, because we want the human gene version of it, but we want to keep the regulatory region from the mouse. So we just add the transcription unit from the human, replace it in a test tube, and we call that a transgene. Transgenes are any intentionally modified gene built in a test tube that is going to be used to study its function when made that way. Homologous recombination is what we need to do, is we want to replace the mouse gene with the human ortholog, transgene ortholog. The morula is a term that we are supposed to learn from early mammalian embryos, which is that we get fertilization, then the cells start to divide, and they first form a morula. And a morula is just a ball of cells that is pre-implantation. And so all these cells in a morula are embryonic stem cells. And so that's what we're going to make when we put together the specialized transgenic cells that have our transgene with a normal mouse embryo. We're going to put them together in a ball of cells, a morula, and then we're going to implant them. And implantation is to put the embryo in the lining of the uterus. So the uterine wall is ready to receive the embryo, and the embryo is going to develop there, and that's implantation. Pre-implantation, the mammalian embryos can be manipulated, but then they need to get implanted into the uterine wall in a host mother. A chimera is what's born. A chimera, you would say, has four parents. Two moms and two dads, and the genes don't mix together. It has separate sets of cells, some from one set of parents, some from another set of parents. And then a transgenic strain is a permanent source of mice that will breed and that represent the original special cell that has the transgene. So it's a strain. That means it can breed true over the generations, and it's this transgenic one that has the transgene homologously inserted to replace the mouse liver enzyme transcription unit with the human. So that mouse will be somewhat humanized. Okay, the last part of our exam, at least the way I'm talking through it here, is this question. Following are found in chick embryo at the phylotype stage. What are these? The notochord. And the key thing about the notochord in vertebrates, they're called chordates because they'll have notochords, and it induces the central nervous system. That's a key thing. They're found in all the embryos, and that's its key embryonic function. Our kentron is just a primitive digestive tube or primitive gut. That's the original embryonic name for our throat, stomach, intestines, like that. Somite 3. So somite is mesoderm that can form different parts of the body, muscles and bones and so forth. And I put the somite 3 on here just for you to add that our somites are numbered from anterior to posterior. And so somite 3 is a specific somite that's up near the head in every vertebrate. Splanktopleura is part of the lateral plate mesoderm that can lines the coelomic cavity in a position where it can contribute to form the heart, the kidneys, blood, and other mesodermal derivatives that become our internal organs. And then the last one is epiblast, and I actually said that word very briefly in class. This is maybe the only point on the exam that's only from the, you might argue is only from the textbook and wasn't mentioned in class. So again, how can you do better on this? Yeah, it's about class and then using the textbook to help understand class. Really focus on the class and bring questions to class, ask questions in class, follow up in class, go home, watch the video again, take really good notes. Because I plan to delete these. So the epiblast is when we see the chicken and it looks like a coin. If we look at it from that view, the epiblast is the upper surface of that coin, and the hypoblast is the lower surface. And the epiblast is going to make basically almost the entire embryo. Okay, and that's it. So we want to go over that stuff. Make sure you got it, and if you have questions, let's get them covered, because this is part of being able to study and prepare, learn, and prepare the second exam, and then remember the final exam covers everything. So these questions will not appear directly on the second, but I might ask some of the same questions or related questions on the final, and if I asked them in a longer format here or a drawing, I might have a fill in the blank and ask the same question. So you really need to understand what's going on here. This stuff won't be covered directly in the for the second exam, but we're building up. So the second exam is going to talk a lot about signaling factors and transcription factors and so forth. Great. Okay. And so we're all set for now.